In this video, we will cover what are the glycans that are expressed on the tumor cells, more predominantly expressed on the tumor cells, and how these therapeutic targets or therapeutic strategies can be developed to target those glycans, so that we can distinguish tumor cells from the healthy cells. So, though we have covered these topics many times in the previous videos, we will cover some new information in this video. So, the figure on the left hand side represents the aberrant glycans that are expressed on the tumor cells that we can see here. Most of these glycans contain sialic acid in it, and some of the known tumor glycans are the siryl TN and siryl T antigen, because of the dysregulations of the C1-GALT1 and other glycotransferase. And what happens because of the expression of more sialic acid related glycans on the tumor cells is that it can interact with the immune cells such as dendritic cells, NK cells, monocytes, and will induce more anti-inflammatory response and reduce pro-inflammatory response. And some of the examples are shown in this side, in this panel, in the middle panel. In this case, what happens is that the cancer cell that expressed cancer cell and that express the lectin receptors such as galactins, they will interact with the dendritic cells or effector cells glycans, that is lacnec glycans, and thereby reduce the effectiveness of this NK cells and the immune cells to act against the tumor cells. So in this case, this aberrant glycosylation helps in preventing this, helps in immune evasion of the tumor cells from these immune cells. And some of the strategies that are developed based on the aberrant glycans that are identified on the tumor cells are presented on this panel on the right hand side. So we will start with the panel A. In this case, the vaccines can be developed against the glycans that are expressed on the tumor cells, and these are more commonly known as anti-glycan vaccines. In this case, we will not only be able to target the tumor cells, but we can also distinguish them from the healthy cells. Uh, Distinguish them from the healthy cells in and since this glycans helps in the immune evasion, so we will be able to target those glycans that are expressed or that are more commonly exposed to the antibodies, and thereby by targeting these glycans, the other antigens will then be exposed to the immune system of the host, and then this will increase the effectiveness on the immune cells against the tumor cells. The next therapeutic strategy is about this antibody immune therapy. So in this case, what will happen is this: glycan binding receptors are being developed, and such that these glycan binding receptors acts on the two ways. First, it will trigger these glycan binding receptors will trigger immune response against the glycans that are present on the cancer cells, and also other way block the glycans. It will block the glycan receptors on the immune cells, such that these immune cells will not be able to deactivate by the cancer cells. As we have shown here, is that these galactins binds to the lacnec of the immune cells and down regulate the function of the immune cells. So this thing can be prevented using this antibody immune therapy. In this ways, we will block the glycan binding receptors on the immune cells. The next thing is the next therapeutic approach is of this antibody immunotherapy. So in this case, the glycans can be genetically modified in the monoclonal antibody in a way that it will this antibody can target the glycans that are expressed on the cancer cells. And this another variant of this antibodies is known as antibody glycosidase conjugate. In this case, this conjugate when binds to the glycans on the cancer cells, it will trim off these glycans. These glycans can be expressed, and these glycans can be are now freely move and can be detected by the immune system, such as this ADCC mediated killing. And when this ADCC mediated killing has the receptor informations of these glycans, it can trigger the more antibodies against those glycan structures. So this will increase the ADCC uh, mediated killing within the host through this antibody immunotherapy. And the next type of immunotherapies is this CAR T cell therapy. In this case, the glycan receptors against the glycans that are expressed on the tumor cells are being transduced into these CAR T cells, or chimeric antigen receptors T cells. So that these T cells now have the now inbuilt have the information about the receptors that are present on the cancer cells, and can directly act against the cancer cells without without wasting times to recognize the antigen that are present on the cancer cells. The last one is about this DC targeting using glycans or DC loading. So in this kind of therapeutic approach is currently being studied in vivo models, where the immune cells or the dendritic cells are being activated by the glycopeptides that are expressed on the tumor cells. And these glycopeptides are being artificially introduced into the in vivo models, so that dendritic cells become activated within a less period of time and can act directly against the cancer cells. So these are some of the therapeutic approach that are currently under research, and these approaches are presently based on the aberrant glycans that are expressed on the tumor cells. If you need to understand more about this therapeutic approach and these glycans present on the tumor cells, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.